Great. So hello, I hope everybody can see my screen. My name is Tomasz Rutkowski. I am from RIKEN-AIP, University of Tokyo, and Nicolaus Copernicus University in Poland. And in RIKEN-AIP, I represent the application side. So my presentation will be mostly about the applications of the machine learning models. I will not talk about the models themselves. So there will be maybe a little lighter talk. So my agenda, a little introduction, then I will talk about the dementia reduction problem. This is the big issue in Japan and the aging societies in general. I will show you some of the examples of our applications and how we try to now move from the supervised learning into unsupervised learning, or maybe even weekly supervised learning, as we discussed yesterday in RIKEN. So basically, we are very open for the collaborations on the machine learning side. And also, I will share some of our non-brain biomarker candidates, our collaboration will work now and what we try next. So this is our team. I am a member of the Dr. Otake Laboratory, CBAT team. So she is our team leader. Those are our members, like usually still in the Zoom connection these days. So we are the laboratory in which we design experiments, we conduct experiments, so we collect the data, we analyze them, and we also apply the machine learning models. So our main goal is to find the neuro-biomarkers of dementia. So we use the EEGs, and there are different devices we have in our laboratory. We run those experiments in the lab, but also we are now preparing home-based experiments. So anybody who would like to collaborate is very welcome, and we always look for the subjects. So our goal is to position our research in the so-called AI for social good. So basically, this is already recognized in the world, and even United Nations put it in the goal number 3 of the sustainable goals, that elderly people really need support, and technology would be really fantastic to improve well-being and maybe delay dementia and all those problems. So there are many negative numbers. The numbers of dementia are growing. The societies are aging. Our bodies are living longer, but our brains have much more issue. There is no pharmacological approach. So all these pharma companies, they bitterly failed and there is no medicine. But as we know from the World Health Organization, the quiet when approaches which are not pharmacological, we are changing lifestyle, changing diet, changing the way we live, and we can really support it with technology. So basically, when we look at the all the subjects on the problems, so around 60 to 70% of the dementia are so-called Alzheimer's disease-based dementia. 10 to 20% are related to vascular dementia, and the rest is like around 10%. So basically, if we can cover monitoring both of those cases, Alzheimer-based and vascular-based dementia, we can really build quite good biomarker. And that's why we focus on the EEG, which is the electrical activity recorded on the skull level. So it's non-invasive method used in the clinics. And vascular dementia for the vascular system, blood system, basically we can use FNIRS. And as we know, there are quite many interventions. So the sooner we can do it, the better for the people. So as Lord Kelvin said already a long time ago, if you can measure it, you can improve it. And we know from the neuroscience that quite many candidates we can use. So basically, there is a lot of background we can already choose. And some of those actually approaches are coming from the area of the brain-computer interfacing. So previously, before I joined RIKEN-AIP, I was involved in the brain-computer interfacing, and I can happily now use those methods now for the biomarkers of the dementia. And additionally, as I said, we also know that the vascular health is very important. So basically, bad vascular health is causes dementia. So that's why we are now developing the system when we can record together, and we have the system already in RIKEN, but basically due to pandemic, everything is a little bit delayed, but we can have also now use collaborations abroad and we can collect data. So the concept is to record together in the experimental setting, EEG electrical activity and the FNIRS. So this is infrared illumination of the skull. And basically we can monitor the blood circulation under the skull level. So basically 2 modalities together will cover around 90% of the possible dementia cases. So basically what is now, and we are a little bit stuck with the thing, so there is a problem with the labels of basically evaluation of people of dementia. So these days, the gold standard is the paper and pencil test, MOCA, MMSE, and so on. So even the latest publications very often refer to those tests. Those tests are very noisy, are very error prone because they are done by the psychologists. You cannot repeat them very often. So basically one score takes for 1 year, and basically brain could change very often. So what we really want to do, we want to use machine learning and completely skip those labels in this case. So unsupervised learning could be one of those our escapes. But let me show you first our results using the supervised learning. So the concept is to build home-based monitoring system. So you can see this elderly lady here watching TV. She's wearing wearable EEG, having iPad, and basically conducting this kind of mental test, which could be done every day even. So basically, like these days, elderly people monitor their health, like, heart health, by measuring blood pressure every day, etc. Now, quite often using different wearables, we want to add in the future this kind of brain tech wearable, which will really show if their lifestyle is improving their well-being, mental well-being, or basically maybe they can make some mistakes, and this will cause, call for some intervention. So the first classical paradigm experiment is coming from the brain-computer interfacing, the so-called oddball paradigm. So basically, we show the series of pictures. Before each series, we ask subject to remember one of them, so-called target. And we showed it in the kind of collection of the random presentation, 7 to 10 pictures. We can also ask subject to make a button press response, and we also collect behavioral responses. So in this case, when we look at the behavioral response, we can immediately see that our subject who are in so-called mild cognitive impairment, so this is kind of beginning of dementia, which we want to pick up as soon as possible to possibly switch any mental intervention, those subjects would have significantly delayed reaction times. But as you will really see, those spreads are quite big, so we cannot use only behavioral responses because they are not very accurate for the classification. That's why we use machine learning, and this was our first collaboration with Dr. Cao and Chivin Cao from our AIP center. And basically, at that time, we applied the tensor train learning to actually see if we can classify those varying responses. So we know from the neuroscience that healthy people will have very nice so-called aha response when they see the picture which they memorized before. People who are elderly and shifting towards dementia will have this response of smaller amplitude and the delay. And when everybody ignores, this response will be quite flat. So basically in the BCI, there's always classification between target, non-target. In our case, we want to distinguish between healthy P300 response and this based related to the dementia or aging. So we review quite many machine learning methods, and the shallow learning method, SVM, linear, LDA, and so on, they basically perform it just above the chance level. So it's not good enough. We use some Riemannian geometry methods, which were also like inspired by Dr. Min from our center, but basically they were around 70%. And the method which was proposed and delivered by the group of Dr. Cao, basically based on the tensor trains, in that time performed around 80%, which is okay for the publication, but it's not still good enough for the industrial or basically healthcare application. So we are still looking forward and trying to push those results much higher, being above 90%. That's why we also tried the Riemannian geometry approach, and this will be the work will be presented next month in Prague. And in this case, we can really see when we use the cross-validation, basically our data sets are not so big still. We can reach around 90%. But when we move back to the leave one out subject cross-validation, which is kind of more honest and the more application, in this case, our results really drop. So the problem is still related to our machine learning methods and basically very noisy EEG. So we also try some other methods. In this case, we try to narrow bound of the EEG, and this was presented in July this year at the conference at the EMBC, one of the larger biomedical conferences. In this experiment, we ask our subject to remember facial expression, emotion expression. So basically, we have short videos of exactly same actor who is showing different emotional expression. And also as a reference, we can also use different actors, so this will be a more easy case. For that one, we use so-called analysis. So this is very famous in physics, but also in the neuroscience, analysis of the brain criticality. So basically, this is using the physical measures. In this case, we use so-called the trended fluctuation analysis, one of this kind of engineered method, which is based on the removing the mean value and then calculating the fluctuation over different time windows. So everything results into a single coefficient DFA, but for the machine learning, we can use those scales, like our fluctuation coefficient over different scale. So basically, if we have 8 channels, from each channel we have 16 scales. So our feature vector will become 128 the dimension in this case. And we know from the physics that actually DFA values characterize the signal as the anticorrelated and correlated 1/f noise, which is kind of the most famous for the resting state EEG, resting state brain. We can have non-stationary and Brownian noise. And what we really see from our subjects our MCI subject, those who move towards the dementia, usually have higher those DFA, in this case called coefficients. So we can tell them that those brains are more noisy. EEG recorded from those brains is much more noisy, much less predictable. But when we go back to the machine learning, we use all the fluctuation coefficients over different scales. We can really see that, again, most of our classical shallow learning classifiers would perform just above the chance level, around 60–70%. The best one would be random forest, and if our shallow fully collected neural network is also above the chance level. What is interesting for us here, we can really see that those pictures, videos we ask our subject to inhibit, to ignore, they are better discriminable comparing to those pictures which we ask subject to attend. So this is really showing that for people who are shifting towards dementia, for them, ignoring stimuli is becoming more difficult and basically more visible in the EEG. So it's very good for us for the machine learning, comparing to those that they are still memorizing. So it's also quite nice kind of neuroscientific results, which we got from the results of the machine learning application. Another paradigm, we call it number 3 now, is called for the related to reminiscent. So basically, elderly people have a preference for the interior subjects who are coming from their childhood. So for our Japanese subject, we prepare 4 sets of the interiors, like Showa era. This is the era from the childhood of our elderly subject. There will be more modern Japanese design. There will be also old Western designs and also modern Western designs. So we also asked our subject to evaluate those pictures. And immediately we could see that our paradigm was nicely designed because our Japanese subjects had really preferences towards the old Japanese designs. They didn't like Western, modern, old, or they were not so also happy with the modern Japanese designs. But what was more interesting when we look at those preferences, like when we put them in the 2 groups of the MCI subjects and our healthy normal subject, we could really see that we have kind of 2 kind of the distribution in this case, that our elderly subject would be kind of more like, bimodal, so they would have positive and negative reactions, while the normal ones would have the kind of more balance. But again, those behavioral responses are not as good for the classification. When we look at the EEG, again, in this case, the data set was not fully balanced, so chance level was around 60-something%. And again, we could really see those results, that those pictures which we asked our subject to ignore, they were the best classifiers. In this case, fully connected neural networks was the best one together with the random forest classifier. And this is our one of the latest paradigms, which we design now in our collaboration with French and German consortium. So we have trilateral project because in the 3 countries, the aging is becoming really a big issue. So in this very fast presentation of the images, one of them again is memorized, shown before the paradigm. And we know that in this case, so we also can collect FNIRS and have different patterns of the blood oxygenation. But what we really see in the EEG, when subject remembers something, they would have this kind of different reactions comparing to the ignored picture. So the blue one would be the reaction when sometimes it's flashing this known picture. When we show control, there would be exactly the same reaction. We can also compare different systems, and our dry electrode system is the best one. So this is our latest purchase. So this one is kind of a little bit colorful, the collaboration, because this data we're collecting now. And we are very much inspired by the latest, results from this wave to vector application. So basically, this is self-supervised learning coming from the Facebook, and they have beautiful this application when they compare EEG or MEG together with the speech. They use it for using nested constructive learning, and then they can distinguish to which speech the subject is listening. We now work on this one to make this exactly same application when we show this video presentation, so we can really use videos as time series, which we will connect with the brainwaves, and hopefully through the contrastive learning, we can classify healthy versus normal, or even having some regression results. So the final experiments we're also running is related to the learning of the new skill. And this is for our elderly subjects. We train them to recognize emotions. And usually we use some semantic description of the emotions. In this task, we ask our subject to learn kind of 2-dimensional grid of the arousal and the balance. So this method is used by the psychologists, but it's not common among usual elderly people. So in first step, we show short video with emotions, then we show where such video is being categorized on this two-dimensional grid, and then we ask subject to put this result. So basically, it's kind of copy and paste learning. In the final step, we show again different series of the videos, but this time we ask them to kind of try to categorize them based on what they learned 5 minutes ago. So this paradigm is testing short-term memory, so-called implicit procedural memory, which is kind of still very strong with elderly people, is preserved in dementia. Also spatio-temporal memory, so this would be a little bit weaker with dementia people, and emotional recognition, which is usually also again preserved in the dementia. So let me show you some results. So again, when we look at the behavioral results over different scores of our cognitive score from our subjects, so that are made psychologists, we can really see some linear fits, but those features are really noisy, so they cannot be very helpful for the machine learning. When we use the trend and fluctuation analysis, so this kind of complexity analysis of the signal, we can really see significant shifts, mostly in the temporal area. So those are the areas when dementia usually starts. We can really see significant differences, and basically, again, in this paradigm, we can see that our dementia subject or MCI subject have more noisy signals comparing to the healthy normals. So again, when we use those parameters for the classification, for the machine learning, and this is for us very nice results because we can reproduce very similar results for the training of the subject and then for the testing. So basically, again, two favorite classifiers, random forest and fully connected neural network, scored about 85% for both cases. And as I will see, those results are reproducible over different paradigms with exactly same subjects. So this is, during pandemic, we couldn't collect any data. We are completely in the lockdown, but luckily with the collaboration with Poland University of Nikolaus Copernicus in Toruń, we could record new data because they don't have any restrictions anymore for the collecting new data. So basically, we reproduced our paradigm. First, 3 years ago, almost, we used it in Japan, very simple wearable. So the results were quite noisy. Now, when we switch to the better wearable EEG, we can really see that our features improve dramatically. So again, we can do machine learning with the simple, in this case, because data is much better now. And we are now preparing back in Japan after the restrictions to run exactly the same experiments. So this is our first step to move into unsupervised learning methodology. And in this case, we try the UMAP, like uniform manifold approximation mapping, which basically is unsupervised clustering. And when we really see for our first data from the low quality wearable, it's very difficult to actually see any separability. So this is like projection of the two first UMAP dimensions. So the blue dot would be for the MCI subjects, orange dots are for our normal subjects. But from the newly recorded data in Poland with better quality, also 8-channel wearable, we can really see that already UMAP can very nicely separate those two groups. So basically, this is very good also pre-processing we can use now for our machine learning steps. So this is very good for results. We also quickly tried using some variational autoencoders, which basically we use for this latent space for our feature extraction. So basically very simple, in this case, autoencoder, having only 2 layers of the input and also 2 layers at the output. So we can train it very quickly. And again, we can really see our noisy recordings, pre-pandemic recordings, are really difficult to be separated. Those are, again, only 2 projections of the 2, like our latent space. But when we look at our newer data, the separability, again, is becoming very beautiful. Again, data set is small, it's a little bit overfitted, but it's really showing that we can really start moving into unsupervised training and maybe use later on our methodology as also research tool. In this case, we do this for the binary cases, normal versus MCI. Next step will be more like regression because we can have different levels of the cognitive scores, and we can really see how this data will be in this case in the regression model represented. But so it's very very nice. And if somebody would have also better idea, we are very open for some new suggestions. So this basically, again, the plot in the 3 dimensions. Again, you will really see our noisy data is really stuck together. Our new data is beautifully separable, so this is very nice pre-processing for our machine learning. So this, what I already mostly was speaking before, it's about the brain waves, so EEG recordings from the brain. But also we try to use other modalities, and this is one of the experiments Dr. Otake, our team leader, is conducting with elderly subjects. Those are the so-called conversational analysis and conversational intervention. So basically, our elderly people are coming to our laboratory to having kind of small conversation, kind of social interactions. So we are recording them with permission of the elderly. Then we can use machine learning to analyze those conversations. So basically, in this case, we are using classical wave-to-wake approach. So basically, we use the pre-trained model to in this case, analyze our speeches. All the speeches are analyzed separately. So for each speaker has own, in this case, vector. And then once we transfer everything into wave-to-wake, we can check what are the topics that each subject is jumping between. And we can really see, we call it conversation pathway, how much within one conversation, which takes around 5 minutes, people are staying on the topic or very much switching between those topics. So this is quite interesting because we can really use machine learning model as an analysis tool for the speech dynamics. So we can really see between different parameters, there are some linear fits. But when we look later how we can predict, like, their cognitive scores, it's becoming quite interesting because we can really see that there would be significant difference between those pathways, although those lengths of the pathways are quite noisy and quite spread. But when we look at our results, our regression, we can really see that we can predict quite nicely ±2 in this level of those cognitive scores based on the speech behavior in this conversation. So this is the work we want to finish and push for the publication. For the moment, it's on the MEDAR high. The other our collaboration also going for this biomarker of the mental diseases and also the dementia is collaboration with the Okinawa Institute of Science and Technology. Our colleagues Zahari Taufik and Professor Takahashi, basically they developed beautiful, very new method that they can analyze the proteomics, so basically the information, what kind of proteins they have in the synaptic clefts from the healthy and living subjects. So previously, to do such analysis, they had to have the brain sample of the subject who already passed away. They could use it post-mortem. In their new method, they are using stem cell technology. So basically, they take the blood samples, they grow new neurons, and basically they can, those kind of artificially grown neurons, they can analyze basically synaptic connection for the proteomics, in this case, information. They can identify up to 5,000 proteins. So it's really beautiful multi-dimensional data. What we really try to help them analyze it with the machine learning in this case, and using also in unsupervised way. This is example from the UMAP, when we try to separate those, for the moment, still binary versus healthy versus schizophrenia, but basically quite nicely showing how we can already implement AI in the very early stages for the research. For them, it's very important to, as quickly as possible, analyze which of the protein candidates are the best for the analysis, because later on, they can search for lower number of the proteins, even they can find 5,000. So this is ongoing, but it's quite interesting how we can quickly plug in AI and the machine learning in the research of the new, in this case, biomarkers. So basically, what next? Sorry, I am going really fast, but we are in this consortium of the German, Japanese, and French, in this case, project. So basically, on the French side, we have INRIA and this Alex Granford, Professor Tonio Bolo from University of Freiburg, and basically we try to develop together let's say international team for the collecting data, but also for the analyzing and providing as soon as possible viable biomarkers for the healthy aging and dementia. We are also now implementing our international, in this case, experiments, which we can conduct them in Poland, Germany, France, and Japan. We have this nice small robot developed by Professor Dr. Otake from our team. And basically, this would be a combination of the robotics, visual stimulation, audio, and also other modalities. And basically, we really hope that this will progress into kind of new development of the easy wearable that everybody can use at home and evaluate mental fitness. Thank you very much. And if there are any comments, I would be happy to answer questions.
Okay, thank you very much, Tomek. So are there any questions?
Questions from EPFL side?
Or suggestions, comments?
I think it's a very important application, yeah.
So yes, let me start from a basic question. So sure. A few pages back, you had the regression experiment result.
Okay, sure.
With the straight, this one. So if I see the data on the right-hand side, so these are extremely noisy
Yes,
And
yes. So
of course, you can take some tendency of data, but prediction may not really be, may not really make sense in this noisy setup. So,
yes, so that's why, like, this is still work in progress, and we really would like to improve it. But basically, you can do it ±2 on this mental evaluation, which is quite okay, because this is as close as quite often psychologists are also making similar errors. Oh yeah, I do agree. Trend is quite interesting, but the results is still not publishable.
But does it really make sense to try to make some prediction from these extremely noisy data set?
Yeah, but practically this is super interesting, because from the speech for the conversational behavior, we can try to predict ±2. So it's quite, I think it's quite good candidate.
So from this noisy data set, we may say okay, one data set is different from data set in some tendency. We may draw this kind of conclusion, even if data is noisy. But if you want to really make some accurate prediction, then already the data is noisy. So then there's no way to have accurate prediction. That's a limitation of the not the limitation of the method, but the limitation of the data set problem.
Sure, I agree. But in the future, we will have multiple recordings. So this conversational behavior, brainwave, and so on. So such, this would be one of the parameters we can use.
Yeah, so you need a lot of effort in improving the measurement system device measurement.
So basically, because this is speech, so this is from the conversation. So this is on a completely different side. We don't use EEG. I believe EEG is the best one, candidate. This is using speech behavior, So this is analysis of the topic of the conversation.
Okay, thank you very much. Any other questions?
May I ask a question?
Yes, of course.
Yes, I'm Kenji Doya from Okinawa Institute of Science and Technology. I'm very glad to know that you have an active collaboration with my colleague. Yeah.
Yes,
and
I'm very happy.
Experiment. So the proteomics from the blood sample of the subject, does it reflect the genetic predisposition of the subject or pathological state of the subject?
I think it's more like pathological state because this is expression of the protein. So this is related to the DNA, but basically based on the expressions, I think it's the pathological state of the subject.
Or even though it is blood, not the brain sample. Still,
yeah, but, okay, but from the blood, the growing stem cell of the neuron. So basically, they have artificial neuron, in this case, synaptic cleft, which is grown from the blood sample.
Yeah, but even genetically twins. So some of them become pathological state, some will stay
sure.
pathological. So environmental effect is also important.
Yeah, but I hope that already in the blood, this maybe is already included.
Okay.
Because so this is a question to my colleagues. Basically, we do machine learning. But they believe that this is quite good, information. Because blood sample is taken already from the maybe genetically modified.
All right. So I asked to Zach.
So ask Zach, he is professional about.
Thank you.
Thank you.